The overall goal of this procedure is to visualize and determine stromule frequency using live cell confocal fluorescence microscopy within leaves, and for visualizing stromules in vitro using chloroplasts extracted from leaves.
These experimental methods can help answer key questions in plant cell biology and chloroplast research by discovering how stromules function.
The main advantage of these techniques is that they produce reproducible and robust data, even of these highly dynamic chloroplast structures.
We had the idea for the chloroplast isolation study because some research suggested that stromule formation required cytosolic structures like the cytoskeleton to form. So we decided to blend up the cell and see if stromules could still form.
To prepare leaf samples, begin by using a very sharp razor blade to cut a small section of an Nicotiana benthamiana leaf. Immediately after cutting the leaf section, submerge it in a 5 milliliter syringe filled with water. Then, remove the air from the syringe and apply a vacuum by using a finger to cover the syringe orifice before pulling the plunger. Gently release the plunger to avoid damaging the leaf section. Then repeat the air removal 2 or 3 times or until the air has been removed and the leaf appears to be deep green. Add a drop of water to a slide and place the leaf section on the drop. Then add another drop of water to the top of the leaf and add a cover slip. If there are any air bubbles, gently tap the cover slip until they are removed. With transmitted light and a